Hello, everyone. So it's my pleasure today to introduce Ed Basham. I'm sure most of you know Ed. This is for his PhD seminar. For those of you who might not know me, my name is Brett Sheffers. I am Ed's supervisor. So I just wanted to give a little background to how I met Ed, just some brief comments in introducing him. So Ed and I met in the rainforest of Madagascar. This was in 2015, 2016. And he joined my research team. We were doing a National Geographic expedition that required kind of climbing into rainforest canopies to survey for amphibians and reptiles. And I only spent a few weeks with Ed during this time, but by the end of my short stay there, a couple of weeks, Ed had stayed on for several months doing work. But just after a few weeks, I had asked Ed to join my research lab. I was a new faculty member at UF at the time and asked him to join the lab. For those of you who know Ed, you might recall that he and his friends have been slowly creating this amazingly complex Dungeons and Dragons universe. Ed and his friends have been working to kind of form this intricate and complex universe of multiple worlds, kind of intertwining storylines, complex characters, plots, etc. And when I had first met Ed and he introduced me to his universe, it had been ongoing for, I think, the formation of, it was something like 8 years going. And so that means now they're in their 13th year. So why am I talking about Dungeons and Dragons at Ed's seminar? Well, I often kind of appear to joke that I offered Ed a PhD in my lab because of the map he showed me in Madagascar. He had this amazingly complex map. And yes, he brought this map all the way to Madagascar. But the reality actually is that I did offer him this position based on that map. And putting his positive attitude aside, he had excellent field skills, climbed trees, did everything right. This map showed me his commitment and follow through, his passion, drive, ambition, his creativity. And I put a premium on creativity and his ability to work with a team. And I truly believe these are the exact attributes that you need to have a successful PhD. And sure enough, Ed carried these attributes over to his graduate research. And I think you'll see today that Ed created a really impressive body of research that tells a nice story, a really nice story with intertwined chapters. So, I mean, in short, Ed made my job very easy. I couldn't have asked for a better student just starting out. He was my very first student coming through my lab, and he has set the bar very, very high for those that follow in his footsteps. So without further ado, I'd like to turn it over to Ed so he could tell you a little bit about the awesome work he's done over the past couple of years.
Thank you very much, Brett. Well, if this was a conversation about D&D, then I'd be much more comfortable, I reckon. But today I'll talk to everyone about frogs. So my title, I hope you can all see that, I assume you can, is The Biodiversity Patterns Across Vertical Height in Tropical Forests. It's a pretty broad title, but we're going to focus a lot on amphibians, but we'll start with the bigger picture. So in terms of biodiversity, this simply means that the biological variety and variability of life on Earth. And in terms of patterns, this is simply the change of that biodiversity across some kind of scale. So it could be some animals here, some animals there, that's a pattern. In terms of scale, we could be talking about space or time. So today I'll be taking a bit of a journey through space and time in terms of the different scales that I'll look at with my chapters. So at the biggest scale, looking at the entire globe, we could see patterns across large scales, macro scales as we call them. So from the tropical forest to subtropical deserts, we could see a latitudinal pattern in biodiversity in terms of composition, in terms of richness and abundance of animals and things like that. That's a pattern. If we zoom into particular biomes, such as this tropical forest, we could also see patterns as well. So just from the top of this hilltop down to the valley, there could be a change in composition and indeed a biodiversity pattern. We can keep zooming in and getting more specific and smaller scales of space. So today I'll be talking a lot about vertical height. And in terms of forests, this can mean a whole huge amount of change in biodiversity from ground to canopy. And these patterns often follow patterns in or gradients in climate, habitat structure, other types of biodiversity and the way things organize. And that's what I'll be focusing on today. We'll also be looking at gradients of time as well, or axis of time. In terms of tropical forests, we see pretty distinct seasons. And in terms of tropical forests, this can mostly be in rainfall. And in tropical forests, which experience these dry seasons, wet seasons, this can cause a reorganization of the communities because of the different traits that they have and the way they interact with the environment and the way that these climate gradients differ between ground and canopy. We can also look at a smaller temporal scale in terms of night and day, where we have, again, climate gradients across the night and day, impacting the organization of communities where the canopy might be more active at night, in the day, depending on the taxa, depending on how they organize. And so we'll be looking at these different scales of space, macro to meso to micro, across horizontal and vertical axes. And in terms of time, we'll be looking at seasonality and day-night cycles, and also the different scales of biodiversity. So we'll be looking at bigger scales in terms of taxa, where we could gather things into birds or bats or small mammals or amphibians, down to communities, single species populations, and even unique individuals. So the goals of today are really to run over my 5 different chapters and look at how these different scales create complexity at different levels and how we can view them in terms of vertical height. So the first chapter will be a meta-analysis looking at general patterns of vertebrates and tropical forests. The second chapter, looking at amphibians. All the next chapters will be about amphibians. This is a community in Madagascar and how they structure. The chapter 3 is in Panama and looking at how seasonality and seasonal climates can impact the vertical structuring of amphibian communities. Chapter 4, I'll be getting even more specific and trying to get as much detail as I can about one particular species and how it distributes across the vertical, horizontal, and seasonal timescales. Chapter 5, using that same frog species, will get yet more specific and look at how its chemical defenses can shift across the seasons due to its vertical movements. And so starting with the bigger picture, we've got the first chapter is the meta-analysis. I actually did this chapter last, but it makes sense to put it first. This was submitted to Oikos yesterday, so fingers crossed. So the objectives here were to really analyze the general patterns of stratification across the tropics. And what I mean by that is simply, where are all the animals? Are they towards the canopy? Are they towards the ground? And do different animals organize differently? Do birds have one kind of organization pattern and amphibians another? We also want to figure out, or at least review the factors and hypotheses for why we might see different patterns between taxa. And we also want to test if there are macro ecological variables that can explain some of this variation that we see between different studies in terms of elevation or latitude. You know, why do different patterns emerge? And so we know that biodiversity is unevenly distributed. We can all see that just simply looking at a tree. We know that different birds occur in the canopy. We see, you know, vultures flying above the trees, and we see other birds on the ground. That's pretty simple. So we've got different animals organizing differently depending on their different traits, and we'll talk about that in a little minute. And we also know that climate and structure is also unevenly distributed. On the ground, you have the ground. You can easily move across it, whereas in the canopy, it's disjointed, it's disconnected. Depending on the traits that you have, it might be more or less difficult to move around. And climate, in terms of climate gradients, the canopy is much more variable. So it gets hotter in the day, it gets colder at night. It's more, it has greater airflow, so it's more drying. And the ground is a lot more buffered. It's got that vegetation above it. It means it's more stable climate, more humid, etc. So we've got lots of individual studies which have recorded where they found different animals across vertical height in different forests, but no synthesis until now. So as mentioned, different taxa have differences in their traits, and we can say their behavior, their biology, ecology, morphology, physiology, endless different things that we can call traits. Just looking at this bat and this frog, we can see that they are inherently different. They have different traits in terms of their morphology and ecology. A bat has wings, it can fly around. It's a huge difference in how it's going to be able to access different resources. A frog has permeable skin, so it might be more impacted by climate gradients than other taxa. But not only do we have differences between the taxa, we also have differences between the environments that they live in. So different forests have different gradients and different structures. So in terms of just one variable, we could look at elevation. A tropical rainforest in the lowlands might have a different structure and different climate gradient to a forest on the top of a mountain. And those different gradients could impact the organization of different animals. So what did I do? I focused on tropical forests because I love tropical forests and they're incredibly complex and diverse. I reviewed the literature and I took data from all these different studies which recorded metrics across vertical height in terms of their abundance, how many individuals, their richness, how many species, and the vertical height itself. I also took those macroecological variables in terms of, was it in Africa or Asia? What elevation was it? What was the latitude, the canopy height, sampling exceeded, and things like that. I standardized the data in terms of bringing everything to the same playing field. So I basically turned everything into proportions between 0 and 1. So everything became a proportion of the maximum value recorded in that study. And that's in terms of the abundance or richness metric and vertical height. And then I employed generalized linear mixed models, where essentially I'm making a whole long list of different models, and I'm trying to figure out which of these variables best explains the variation in the data of why some studies show animals going up towards the canopy and some show the opposite, or vice versa, and what best describes the variation. I then take the best models and take the predictions from those models and average them together based on how well the model performed. So we're using our best models to make our predictions. This is the distribution of data points that I had. So very simply, we have a lot of data here collected in Central and South America, not so much in Africa, a reasonable amount in Asia. We had lots of studies from birds, quite a few from bats, not quite so many from small mammals, and only a few from amphibians. Most of this research spiked in after the 1990s, where small mammals and amphibian studies started to pick up. In total, I had 62 studies, and I could break these into 86 different locations, which I called study units because some recorded different sites in different countries, so recorded them separately. So this is the main figure and the results that we got from this study. So on the y-axis, I have all my different studies here in terms of just abundance, and I have my different taxa. And on the x-axis, I have what's the slope coefficient, essentially, of if it's to the left of this line, has a negative coefficient, that means the animals, the abundance in this particular study was declining towards the canopy. So there was more animals towards the ground than there were in the canopy. If it's to the left of the line, to the right, there was more. And so we can start to look at the individual taxa and the variation. In bats, we saw a lot more of studies were to the right of the line and very significant. So there were generally more animals towards the canopy in bats. In birds, it was reasonably variable. There seems to be a kind of leftward trend, but also we've got some more significant on the right, so a decent amount of variation. But in small mammals and amphibians, we had these always declines, so consistent declines in terms of their abundance towards the canopy. We can also look at our species richness, which was using a slight different data set, but essentially the same. It's the same analysis. And we've got very similar patterns, so variation in bats and birds and univariate declines in small mammals and amphibians. Now, why is that? Now, we've used our macroecological variables to try and derive these predictions, and I can talk about that in a minute. But in terms of the traits and the reasons why these taxa might do what they do, we can just about see straight away that there's the flying animals up here actually have some of their highest abundances and diversities in the canopy. And this might make a lot more sense because they can simply access those resources a lot easier. They can become specialized to using those resources and move around. Whereas the small mammals and amphibians, they can't necessarily move around so well in the canopy. And In terms of small mammals, they're generally generalist feeders, so they can feed on nuts and seeds and fruits and insects and whatnot. And all of those things, whilst in the canopy, sometimes fall down to the ground. And just think, here in Florida, how often do you see a squirrel on the ground? Even though it's arboreal, it's probably collecting food. So foraging is actually probably very important for the stratification pattern we see here. Probably not for amphibians though, because arthropods that they eat occur all the way from ground to canopy. And it's probably that climate gradient that actually influences them and means that that buffered gradient, that buffered climate on the floor is really beneficial. They can breed around, they can live a lot easier and diversify, etc. So I haven't actually, that's not part of the analysis. I'm just bringing in some of the main citations that I found in this analysis, but we can at least see here the general patterns that we have. In terms of the macroecological, I'll just break this down. It's very simply, it's the canopy height we have as a macro, elevation and latitude. We did actually use more. I'm just presenting these three against our species richness and abundance patterns for our different taxa. And just so that we can really easily understand this, I'll highlight the main takeaway. Bird stratification was negatively correlated with elevation. And that was our main result from all of this analysis in terms of macro. Elevation, bird stratification has been shown to follow foliage density. So the more foliage in a particular strata, the more birds you expect. And we know that foliage density is highest in the canopy of lowland rainforests, or highest in the understory of montane rainforests. So what this is showing is that more birds are weighted towards the canopy in the lowland and more weighted towards the understory in montane forests. So we have a pretty good hypothesis and link to why we're seeing this pattern in vertical stratification change across macro space. We didn't see many other patterns in our other taxa, but it might be because of data scarcity. So just to look at elevation here, we had a really good number of bat studies, but not many that actually sampled bats above 500 meters. So can we actually analyze that pattern? Not really. In terms of small mammals, also quite limited in terms of elevation. Amphibians, we have good coverage, but away we've only got 6 or 7 studies. So actually trying to analyze a more complex macro variable doesn't quite make so much sense. So this study has really found that we've got some missing data gaps. So what does it mean? We've seen really significant variation both between and within taxa. And the most important was really what taxa are we looking at? And that gives us most of the idea of what we're going to see in terms of stratification. There are obviously scales and environments that haven't been well explored. And we can tell that there's, or at least I'm going to go into the fact that hidden complexity exists within each of these studies. So we've really condensed everything down. You know, all of this nuance that might arise from the various different species, the various different environments that these studies are conducted in, there's a lot of variation there that we're missing. And so just to look at the amphibian studies here, Sheffers and Basham, who's that? Basham 2019, this is me, and I can tell you there's a lot of variation in here that we're not quite seeing because we've had to condense it down just to enable ourselves to look at general patterns. So that's what we're going to do now. We're going to break some of these down. So in these subsequent chapters, the main methodology was to climb trees and find frogs. This is me up a tree in Panama. I didn't find a frog right there, but I did find a frog just about over there. This is hanging above a stream. What we did is structured vertical surveys from ground to canopy, that's 0 to about 30 meters up. We caught all the frogs that we could, noted them down, and sampled across lots of different trees in our environments. And that way we can figure out where frogs live in terms of their vertical height. So in terms of Madagascar, vertical stratification of amphibians in Madagascar, this was published under a slightly different title in the Journal of Animal Ecology in terms of its distance decay relationships in horizontal space. If you want to check that out, give it a read, but I'm just going to focus on a different aspect, a more simple aspect of this study here. And so just exploring how the community changes vertically in terms of its composition. So we're adding some extra complexity here, looking at composition, its richness and abundance. And also looking at how shifts might change across night and day. So what did I do? This was the site in Ranamafana National Park in Madagascar. I sampled all these different points, our different trees, ground to canopy surveys for 70 trees and 140 ground to canopy surveys. And it's a lot of work. It was a good 3 months. So what did we find? What I have here is a Venn diagram of the number of species that were found in different strata. So I have my ground, understory, and canopy. The ground was 0 to 1 meters, understory 1 to 4, canopy was 4 to 20. So we can see there's a pattern in stratification of richness. We've got 13 species only occur on the ground compared to 4 that only occur in the canopy. And there's generally a lot more species on the ground than in the canopy. This is that same data that went into the meta-analysis, essentially. And we've also got a pattern of species turnover across vertical space. So here, what we have are similarity measures between our different strata. It's called Jacquard similarity, where 0 is dissimilar, 1 is similar. Ground against canopy, very dissimilar. Ground against understory, pretty dissimilar. Understory against canopy, pretty similar. So the cool thing about this is that we're actually seeing a really, really rapid change in terms of composition just from the ground to the understory. So that 1 meter difference is making a huge difference in terms of the change in composition across height. So in the meta-analysis, we saw, okay, there's a decline, but here we can see that that decline is not even across height. And in terms of abundance, what I have here is a density plot against vertical height. Essentially, what was the density, the number of frogs that we found at different heights. And we have split by the day surveys, AM, and night surveys, PM. In the day, frogs are more active on the ground. So you can see this black line is peaking here towards the ground, which makes sense because like we said, the ground is more buffered and the sunlight, the heat doesn't quite reach down to the ground in the day, so frogs are still able to stay active. Whereas at night, yes, they're still mostly occurring, there's well, at least greater abundances on the ground. We're seeing that decline that we saw in the meta-analysis. There is also a lot more activity up into the canopy, and that's when the climate has become acceptable for a lot of these creatures to come out, and moisture is rising from the ground, there's mists in the trees, so it's plenty humid and not too harsh. But both of them, as I say, both in the day and night, still patterns of more abundance on the ground, and that's that decline that we're seeing into the canopy. So what does it mean? The canopy and the ground are inhabited by different sets of species. The decline in their abundance and richness, and indeed their composition, is not consistent with height, so it's not entirely even. And we can also see that the spatial pattern in distributions are differing between night and day, or at least in terms of activity. So moving on, we have vertical stratification collapses under seasonal shifts in climate. This was published in the Journal of Biogeography. Please check it out, give it a sight. In terms of objectives, we're looking at the seasonal vertical movements of amphibians, and we're trying to get more specific. So look at specific species and what everything does and break down the community. And then we also want to consider these seasonal patterns in context to climate change. So what did we do? This is a similar map to before, but this one is in Panama, my different trees across my site. The site is here in central Panama, close to Panama City. And we're looking at seasonality. So we've got our rainfall here, and I can show you here that we've got a dry season, January, February, March, much drier than the rest of the year. So we did 2 sets of seasonal sampling. We did our dry season sampling, we did our wet season. And for 45 trees in the wet, 43 in the dry, 66 surveys in the wet, 55 in the dry. And I sampled the same trees across seasons. So we have that replicability of sites. So in terms of results, we're trying to figure out which species live where and do they shift vertically. Here I have my wet season community and my dry season community. And instead of last time where I broke the strata into 3 kind of distinct categories, what I've done here is just broken down vertical height into 1 meter intervals. Each color represents a color-coded species, and each box represents if a species was present at that height. So here in the wet season, we've got that, again, that rapid decline in species in the understory, so it's not even with height like we saw in the meta-analysis. But then we've got more boxes, more colors up in the canopy in the wet season. So we've actually got new species that we didn't get on the ground are occurring in the canopy in the wet season. So it's a complex environment up there with interesting species using the canopy microhabitats. Whereas in the dry season, we've got, again, rapid decline until just 3 or 4 meters up. We've got this huge decline in species. We've only got 3 species occurring and no new species. So we've just got this complexity is declining in the dry season. And where are these frogs going? They're possibly descending towards the ground because, as I was saying, that canopy environment is getting a lot drier during dry seasons. So because of the heat of the sun, the epiphytes in the canopy will dry out, and if they dry out for long enough, then those frogs are simply unable to stay there if they can't hide away and estivate, and they'll have to come down to the ground, which is climatically buffered, at least more so than the canopy. So in terms of abundance, I did a randomization approach, and that's why we're going to see a histogram of distributions where I got the mean heights and compared the mean height of the total community in the wet season versus the mean height in the dry season, and I created a differential. And so I can see how far they shift between different seasons. On average, the total community of this forest declined by 5 meters in vertical height. But in terms of specific species, we can see all kinds of different patterns. So this was one of our biggest movers, Bristolanthes crentus, and it moved 13 meters on average. So a pretty huge decline in terms of where it was in the forest. The biggest mover was Andinomatus fulguritus, which moved entirely 26 meters, and its entire population was up, living in the epiphytes in the wet season, and then to come right down to the ground, living in the leaf litter underneath the roots of big trees in the dry season. So we've got species-specific patterns, and also, you know, I've only showed you 2 species here. There were a number of others. There were some that only moved, say, 1 meter down. There was others that moved only 0.5 meter down, and there was one that actually moved up 0.3 meters. So there are different frogs that do different things. And we're seeing here that fulguritus evidences a migratory behavior because it's actually moving in a seasonal pattern between breeding resources in the canopy and non-breeding habitat on the ground. So what does it mean? We can see that there's so much flexibility in these systems. This community isn't static. And here we're looking at seasonal timescales. We've already seen from the previous chapter that they're flexible on night and day. And there's a lot of variation between species. So we keep seeing more complexity. And we can also bring in climate change here. And when we're looking at climate gradients in general, if we can link them to what we expect climate change to do, we can start to make predictions. And in terms of this community, climate change is going to, or at least predicted to, increase the strength and length of droughts across tropical regions. So if that occurs in this community, then we expect that all these frogs will be forced down to the ground for longer. And that could lead to a whole bunch of knock-on effects in that community. So it could be more predation due to ground-based predators, it could be more competition for resources. It could be increased disease prevalence because actually research has shown that chytrid fungus, which is decimating amphibians, is actually more prevalent on the ground where it's more moist. So there could be, you know, increases in disease. There will be some species that benefit. There's always going to be some species that do better than others, but overall, it seems that the majority of these species will likely be negatively impacted in terms of their vertical position by climate change. So we've got a lovely frog getting forced down to the ground. So can we get more specific? Of course we can. This is my cool yellow frog that I found 24 meters up, and I'd been searching trees, you know, all through this field season, and it was one of the last trees that I found, and I just saw it and was like, this is a monster, absolute gigantic tree, really unique. And I climbed it and I find this incredible frog, the first one I'd found in that field season. So instantly right there, I was thinking, there's got to be something in this. This is pretty cool. So this whole chapter is dedicated to that relationship. So titled Large Old Trees Constrain the Vertical, Horizontal, and Seasonal Distributions of this frog. It's in revision at Ecology. We're trying to assess, and so it's an arboreal frog that lives on trees. And so we're trying to assess which tree characteristics drives its distributions in terms of vertical and horizontal, and indeed seasonal, and investigate its phylopatry and migration, and if those things occur. So phylopatry is simply the tendency to stay at or return to the same location. These are frogs in the dry season in the leaf litter, and then up in an epiphyte in the canopy in the wet season. What did I do? I surveyed for these frogs quite a lot. So 3 dry seasons and 3 wet seasons, 107 trees in total. So 41 were Espave, and that was that first tree that I found it on. And I thought, maybe there's something special going on. So I targeted this tree specifically, whilst also sampling a whole bunch of other trees to get a better look at the entire community of this forest. So 66 trees of 37 other species, over 200 ground to canopy surveys, 400 ground surveys, lots of effort. We took photos of each frog, and they have unique patterns on their stomach, so you can tell who's who, and you can track them where they go if you find them again. And I recorded habitat variables from each tree because we're trying to figure out which variables are important to this frog. So we found pretty quickly that Espave is a very unique tree in this forest. And so what we have here is a principal components analysis, where essentially you're comparing the different trees against each other based on their characteristics. And the further away the points are, each point is a tree, the more dissimilar they are in terms of their characteristics. Espève here and all other trees over here, we've got an incredible separation in terms of their characteristics. Espève are bigger, they have more roots, they have more epiphytes, they have bigger canopies, they have more moss on their trunk, etc. So they're a pretty cool tree in this forest. This is my favorite tree. This is where I found that first frog, just there. And actually, my brother might recognize this because he climbed this and that might have been the rope that he used. So I applied these generalized linear mixed models again, here using a different binomial distribution, asking what habitat variables best explain why they live at certain trees. We found that DBH was simply the best predictor. So we have diameter of breast height here against probability of presence of the yellow frog. The bigger the tree, the more likely you are to have this frog. But then when we break this down between non-Espève and Espève, we see that DBH is really important, but Espève are providing all of the big trees, or at least most of the big trees in this forest. So it's hard to tease apart, does the frog love Espève or does it love the characteristics that it provides? And those happen to be unique in this forest. So we also know that DBH was closely correlated with how big the roots were and the epiphyte density. And so these frogs actually need these epiphytes to breed in the wet season. So it makes sense that they are huddling around these trees, these massive trees that have all the resources that they require to live. And so we've got our distributions across space in terms of their horizontal and vertical. We know that they like big trees, but maybe big trees don't just occur everywhere in an even distribution. We might have something like this, where you have trees across the landscape. And if these frogs need these resources so much, maybe they don't want to move. And so we're thinking, do they actually move between trees? Like, how do they find new trees? All these kind of questions we want to try and ask. And we've also got their migratory pathway, which we've already looked at, but now we have a bit more data to try and confirm this. So we measured how far away each frog was on the ground when it was in the dry season, and how far it was away from the trunk. So here on what we have on the left here is simply the count of frogs in the distance away from the trunk. We have a real easy decline, and in terms of the actual density of frogs per meter squared, a really sharp decline. So we can see that these frogs are actually huddling right around the trunk of the tree, because why would they want to wander off into the forest if there's no beneficial resources for them? They want to stay where they are and keep using those epiphytes in the wet season. And as soon as it starts raining, they'll start climbing up that trunk and going back up. And so here we've got more data. We can show that the dry season against height here, all of these frogs pretty much are found right on the ground, versus the wet season, mostly, almost entirely up in the canopy, up in the epiphytes in the wet season. So we also did our capture mark recapture, as I mentioned. We had 370 captures and 200 recaptures. Our 200 recaptures, 98% of those were found at the same tree, and that was regardless of across seasons and years. So we know that they're staying at the same tree. I found some of these frogs so many times. I'd come back 2 years later and I go, I found you, I've seen you again, and he's at the same tree, going up and down. So I found him on the canopy, then down on the ground, then back in the canopy. But we did see that 2 frogs moved. So we had one movement of 63 meters between a tree, 63 meters away, and a 204 meter movement of a frog. So this is a 14 millimeter frog. And this is a tropical forest with streams and fallen trees and everything. And he's managed to move 200 meters to a new tree. So this is really important because it shows us that they do have a dispersal pathway. They can move around, they can seek new habitats. Of course, these trees do die eventually. They have to find new trees, so they need a way to do that. But nonetheless, there's a really strong pattern of adult pholopat trees, so they're staying where they are. And why wouldn't they? They've got all of the epiphytes, all of the resources they need. So what does it mean in terms of conservation? We can see here that DBH, the biggest trees, are really important, and especially for this species. Here we've only looked at one species, but big trees provide unique resources in every single forest in the world. And it takes a long time to replace these kind of trees and the habitats that they provide. So we keep zooming in. We've got this really specific frog here, but still we've got this incredible complexity. And we've shown that this frog is doing a micro migration across 30 meters of vertical height, and step aside wildebeest, because wildebeest, they move across thousands of miles of Serengeti, And this frog is doing exactly the same thing in a pattern that we see across great landscapes, but across 30 meters of vertical height. So we could get even more specific. We could look at the individual epiphytes that they're using to breed, and we could look at their tiny territories on the branches of each tree. You know, we could keep breaking it down, and it would be really cool. That would be a great thing for future research. But have I missed the coolest thing about this frog? Yes, I have. How could we forget? It's poisonous. So chemical defenses shift with the seasonal vertical migration of this frog. So using the same frog, the same system. This was published in Biotropica. Check it out, give it a sight. So we're looking at its changes in defensive compounds across the seasons. And then we can bring in climate change again and try and make predictions on how climate change might impact this species. In terms of themes, this chapter has a lot of really cool themes that kind of interact with each other. So we first have its toxicology. So we have the alkaloids that it uses to defend itself. We have its movement ecology. We have the fact that it goes up and down between the seasons. We also have its diet. And contrary to popular belief, these frogs do not produce the alkaloids that they use themselves. They actually eat arthropods and take the alkaloids when they're digesting them, sequester them, and put them into glands in their skin. So it is the arthropods that they're eating that are super important for them. And we also have their movement ecology is therefore influencing their diet, because on the ground, there's a whole bunch of different, well, vertical stratification again. We've got different insects and different arthropods occurring on the ground versus in canopy. So it goes to show that their diet is therefore going to influence their toxicology. They're eating different things in different places, getting different poisons. We've also got climate, because as we've shown, they move up and down with the seasons. And that influences their movement ecology. But not only that, different seasons also impacts the abundance and composition of arthropods available in the forest. So we have a kind of double whammy here. So what did I do? I took 26 frogs, 13 from each season. We extracted the alkaloids, we gave them a little buzz, collected the alkaloids, and then released them again. They were fine. We ran these alkaloids through gas chromatography and mass spectrometry. So we use these sci-fi machines, and big thank you to Dr. Ralph Saporito, who was instrumental to this research at John Carroll. We then use these machines to basically blast the chemicals with electrons and figure out what the structures are. And from doing that, you can assign the curves and weights to enable you to quantify how much alkaloid was in each frog, and also pinpoint what kind of arthropod these alkaloids came from, because different arthropods produce different structures of alkaloids. So if you know what the structure is, you can make a pretty good estimation on where it came from. So what did we find? We found that in this frog, they're eating a lot of mites, and they're actually getting most of their alkaloids from mites. And so mites and ants were the most important. So nearly every alkaloid we found could be attributed to ants and mites. And this is sort of contrary to the general view of dendrobatids in most literature in the past. It has been pointed to ants as being the most important. We're now showing, and this is really good extra evidence to show that mites are also really important and could be the most important for some species like this one. We also saw that they had more alkaloids in the wet season. So here we've got our blue bars, wet season, higher than the red bars, and this is a significant relationship. We didn't see this pattern for richness, which is not entirely surprising because they can hold on to very small amounts of each alkaloid for a long time, so we might not see any change in richness. We also saw a change in the composition. So again, we've got similar to that PCA, this is an NMDS. We have our dry season frogs in red, wet season in blue, and we have a clear separation due to the composition of alkaloids between the two communities in the two different seasons. So we know that they're getting different alkaloids in their different seasons, and this is almost certainly because they're eating different things in those different environments. And we also know that having more types of alkaloid makes you more defendable, so it's better for you. So what does it all mean? We can tell that mites are really important alkaloid sources for dendrobatids. We can see that climate change may actually lower the defensive potential of this frog. So, I mean, this is only a preliminary study, but we've already shown in previous chapters, already suggested that climate change, if it lengthens and strengthens droughts, and this frog is having to stay on the ground for longer. Well, here we showed that if it's staying on the ground, it might end up with less poison, because we found them with less poison, less alkaloid in the dry season. So we can also see that these movements across vertical height that this species does, not only are they tied to their life cycle processes in terms of breeding, because they need to breed in the epiphytes above, but they're also gaining defensive potential by eating the different arthropods that live in the canopy versus on the ground. So taking a big deep breath, we've looked at a lot of different scales and a lot of different types of complexity and different types of biodiversity. And we can see that this complexity exists at all scales, but in different ways. We see different patterns. You know, when you look at this macro scale meta-analysis, we've seen the generality of patterns, but then we've got super specific, and we can see that a particular frog changes its poisons depending on where it moves up and down. We've made a really good study of amphibians here, and we've seen that they present really strong patterns of stratification in terms of their richness, their abundance, their composition, etc. And that we've shown that this stratification is not even across height. And temporal variation is often linked to climate, which for amphibians is particularly important for them. And indeed, we can start making suggestions on how climate change is going to impact these communities and start restructuring the actual vertical organization of communities and have big effects. And remember, every single forest has a ground and a canopy. So research regarding the vertical gradients of height, the vertical gradients of forest, can be applied across all of the forests that we have. And so research in this field, I think, is really important, especially as it's a neglected science, as it were, because no one really wants to climb trees. We've also found that fitting 5 chapters into a single presentation is not advisable. Don't do it. Thank you very much, everyone, for listening, and a huge thank you to Dr. Brett Sheffers, who has been instrumental throughout this research and just a great advisor. Thank you to all my funders, all my great people that have helped me in the field. My family have been great, and just an awesome time. This is the picture of me and Krista and the team in Madagascar. I thought this was pretty funny because I look so young. It's ridiculous. And yeah, time for a holiday. Thanks very much for listening, everyone, and feel free to ask questions. If we don't get to your questions, feel free to send me an email.